part of the oncology conference, we'll try to keep our conversation limited to mostly to your progress in the IO for the IO programs. And maybe to kick things off, can you give us recent developments or any recent updates for your lead CAR T assets, and what investors should primarily focus on over the next 6 to 12 months in terms of regulatory updates as well as clinical development?
Yeah, thank you, Kalpit, for having us at this conference. It's our pleasure. Oncology is one of the main pillars for CRISPR Therapeutics. If I just, before I go into our CAR T programs, just take a look back at the last 60 years or so of our fight against cancer. We've made progress, but still there's a lot work to do. We started with a bunch of small molecules, put them in different cocktails. A lot of them were very toxic, and oftentimes cancer care was associated with significant side effects. We then had the era of targeted antibodies, and we've obviously seen a lot of benefit from those targeted antibodies. We've seen the advent of personalized medicine, but still, I think it's hard to find cures in oncology. A lot of times, the medicines that we use have significant side effects, leading to a very poor quality of life. And I think now that's where we're entering into this new era of cell therapies as targeted agents to fight the cancer, and do it in a way where oftentimes it can be single dose or a single course of treatment with very limited side effects compared to what we see traditionally in oncology. So we're quite excited for the next 10 to 15 years in the development of cell therapies for various oncology indications. And we hope to be at the vanguard of it. For us, this is a fundamental investment because I think cell therapies can only be successful if there's sophisticated engineering. And the best way to engineer them with a sophisticated tool is to use CRISPR. We've now seen cell therapies and our cell therapies going to the clinic now with 5, 6 edits. These are quite sophisticated constructs, and they're only going to get more sophisticated as we discover new ways of making these cell therapies more potent, more durable, and even safer. So that's sort of the preamble for our investment and our continued efforts in oncology. Where we stand today is that we have efforts ongoing across 3 different targets: CD19, CD70, and BCMA. In CD19, what we disclosed were data for our CTX110 asset. This is an allogeneic CAR T targeting patients with relapsed and refractory lymphomas. And for those who are not familiar with allogeneic, we take a healthy donor blood sample, we extract the T cells from it, and from those T cells we make CAR Ts. And these are immune cells targeted to kill the cancer in our manufacturing facility, but done in a way where it's done across patients. It's not individualized. And these cells can then be administered to patients right away within a couple of days of diagnosis. And some of them have shown remarkable effect in terms of complete responses. So what we showed for our CTX110 program is a relatively competitive profile. We can talk more about the data, where patients with a single course of treatment or single dose have been in complete response for over 2 years. And we showed that 1 in 5 patients, or 19% of the patients treated with a single dose regimen, in some cases or optional redose, had durable complete response. And that's quite significant for the first effort in allogeneic CAR Ts that could move into registrational trial. We then have the next gen CD19 CAR T in CTX112, where we hope to dose patients this year, and we'll have data, but this is optimized for potency, and the data could be even better. In the interim, though, however, I'll talk about a registrational trial in CD19, which is the 2-dose regimen, which could also bridge us to a much better efficacy. CD70 is a very intriguing target. There are a number of pharma companies that have worked on it before. It's never been the right balance of benefit and safety in the ADC setting or antibody setting, but in the cell therapy setting, what we saw were pretty encouraging data both for T-cell lymphomas as well as solid tumors. We've probably demonstrated the world's first complete response in a solid tumor with an allogeneic CAR T when we presented at the SITC conference last year. And we can continue to do work to improve the potency of CTX130 in the form of CTX131 targeted towards CD70. In the meantime, we're trying to move forward as quickly as possible with CTX130 in T-cell lymphomas, which hopefully gets to market as soon as possible because these patients have no other options. I know that was a long description of our portfolio, but we're encouraged by both the near-term assets, CTX110 and 130, in CD19 and CD70 respectively, and then the next-gen assets we're very excited about.
Okay, that's a helpful overview, and maybe I want to dive deeper into some of the things you just mentioned, starting with the 19% 6-month CR rate that you saw with CTX110. I think the pulse I'm getting is some investors might view this as sort of a half glass full, and some might think of it as half glass empty situation. The 19% might not be enough to get investigator interest, but then some might say it's better than 0%, right? These patients have essentially no option at the late line setting. Curious to hear your thoughts on this. How competitive do you think the 19% would be if you receive approval?
Well, one of the things we've noticed, is the excitement around enrollment in our trial remains very high. And that's always an indicator of how much physicians believe in this therapy. I think they've seen the data, but there's, the number of sites that want to dose patients tomorrow with CTX110. The reason being, while there's data from autologous, and again, you have to look at apples to apples, an ITT basis, autologous therapies are probably at about 30% durable CR rate. But that's very SCARDA, and that comes with a significant proportion of the patients getting CRS and some patients getting ICANS in the real world. And what we've shown is, you can dose patients 2 days later. That was the mean time from diagnosis or enrollment to actual dosing. And we can, you can see durable responses in patients with the healthy donor-derived allogeneic CAR Ts. And 80% of patients in the U.S. are cared for in community settings. A lot of these patients have no access to autologous therapies. Many of them, don't want to come in for bispecifics over 9 courses of treatments because they have to travel to their site of care. And if you have a benign, relatively safe allogeneic option, where the practices don't have to deal with CRS and ICANS, and they can administer in an outpatient setting, there's a huge market for this. And right now we're at 1 in 5 patients, you mentioned 19%, getting durable CRs. With our consolidation dosing regimen, where we have 2 doses instead of 1, I think the number's going to be even higher. And so we're very much in autologous territory already with CTX110. And we have the agreement with the regulators now about what the design and requirements are for the registrational trial. And so we're all systems go on getting CTX110 to approval. And we believe this is going to be a very competitive drug.
Okay, and you recently had discussions with the regulatory agency and opted in to do a pivotal study with consolidation dosing at dose level 4, and combining that with standard lymphodepletion. I guess what went into the decision making for consolidation dosing versus a single dosing regimen?
Yeah, a few things, which is we saw in our Part A trial that repeat dosing helps. There are patients where the tumors start growing, but then you have repeat dosing and you see tumor shrinkage or tumor elimination. This is because you have a ratio that's really important of effector cells, which are the CAR Ts, to the tumor cells. So the more effector cells you have, the more likely you are of getting rid of the cancer cells. Right? What we saw is that we can do repeat dosing in a relatively safe way, even if you have to do LD chemo again, and patients are able to tolerate a second dose without any change in the safety profile of the drug. And these are data we haven't shown yet for our Part B of the trial that had consolidation dosing. We saw conversion of patients that had a PR after the first dose to a CR after the second dose. We saw instances where they had a CR in the first dose that led to a deepening of the CR of the second dose. In other words, the patient again MRD negative essentially, and in fact even stable disease turning into a CR. So so there's definitely a benefit with the second dose because again, if let's say you had 1,000 tumor cells, you brought it down to 100 after the first dose. The second time around, the ratio of effector cells or the CAR Ts to the number of tumor cells is significantly higher, and your chances of getting to complete response are that much greater. And so we feel confident that the consolidation dosing will have an improvement of the efficacy while maintaining the safety profile. And that all of a sudden, makes it an even more competitive profile from a market standpoint.
Okay. And can you remind us if that pivotal study is lymphodepleting patients again before the second dose?
Yes, we do. But mind you, our lymphodepletion regimen is a standard lymphodepletion using autologous therapy. It's not very high or enhanced lymphodepletion that some other companies are using. We believe that the whole thesis is this is a product that's going to take off in community settings. Several cancer care networks that want to use it. What you don't want to do is create a safety profile for the drug that they can't manage. And I think enhanced LD, whether it's in the form of higher doses of flu-cy or addition of other agents like Campath, I think will just make it a less competitive profile in community settings.
Okay. And I guess, when you do give that second round of lymphodepletion, do you think that sort of limits the total pool of patients that would be eligible for CTX110? I think I get a pulse from investigators that they're sometimes scared to lymphodeplete these patients again, right? Or there's a group of experts that don't want to do this the second time. Curious to hear your thoughts on that.
Yeah, I think so far, some of these reactions come early on before people see the data, right? Most people assume that the second LD regimen would, cause more infections or prolonged cytopenia or anything else that we might see. But what we've seen is that we don't see any of that in the patients that we've dosed LD chemo again 30 days later or 35 days later. There are patients who progress very rapidly and are very frail, and they won't get the second LD chemo, right? I think it's just anybody who shows response and benefit, I think will be eligible to get the LD chemo, and a lot of patients will have to take it. I think compare this to other approved therapies. R-CHOP is not a walk in the park. People when they take the first course of R-CHOP, they have significant talks associated with that. Any approved therapies, it's not easy. But then if they see benefit, they continue on because that's what patients want to see, is open an opportunity to get a complete response. So that's something that we don't think is going to significantly impair or handicap the therapy. In fact, it's only going to make it more attractive.
Okay. Talk to us about CTX 112. I guess, what are you hoping to achieve by introducing this next generation product of allogeneic CAR T?
Yeah, we've taken a strategy that, we don't need to improve the persistence of these CAR Ts for very long. I think the CAR Ts do their work, within the time frame of 10 days or so that they're in the patient. What we want is the CAR Ts to not get exhausted in the tumor or in the tumor microenvironment. So we did a massive large-scale screening of T cells to see what's going to improve their potency. And these were pairwise screening. So it wasn't just one edit, it was multiple edits to see what combination works best. And we came upon this combination of Regnase-1 and TGF-beta-R2. TGF-beta-R2 is slightly better known in the field, as others have also gone down that path of knocking out TGF-beta-R2, because TGF-beta suppression is a major way the CAR Ts get exhausted. But then Regnase-1 is a novel target. And we see both in preclinical data and in manufacturing that these cells are much more potent. So even if you have the same window of around 14 days, let's say, that these CAR Ts are able to do their work, what you're going to see is, much better potency in my mind in patients because the cells are engineered that way. Now, we have to do the trial and make sure it's safe and you don't get CRS or any of the other things. But we're quite excited to start dosing patients with CTX 112 and 131.
Okay, good. I want to move on to CTX 130. You have two indications that you're developing for this CAR T. It's T-cell lymphoma and then renal cell carcinoma. I guess what's the benchmark here in each of those indications in the salvage treatment line, and how does your data fare relative to what's used today?
Yeah, T-cell lymphomas is sort of an area that there hasn't been much development. People are still using agents from 15, 20 years ago that were approved, on a conditional basis. We had NPR did a story on one of the patients that were treated in our study recently, independent of us. And it was remarkable. This patient had tried, several lines of therapy, kind of given up, and all of a sudden gets a complete response. If you look at the benchmarks in T-cell lymphomas, complete responses are unheard of, and even the ORR is in 30% range. So these are patients that basically have no other options. In a small sliver of patients that are CD30 positive, you can use brentuximab, but then it comes with significant neurotoxin and everything else that goes with that therapy. So what we're looking at is what investigators think is a revolutionary way of treating these T-cell lymphomas. And I think as we optimize the dose, as we optimize the regimen, we're going to see, hopefully more patients get into durable response with CTX 130. So we're discussing, we're having discussions with regulators now to figure out what the pivotal or registrational trial might look like for CTX 130, what's required in terms of patient numbers, and what the benchmark is. And so we'll have more guidance on that in the near term. But we're excited about CTX 130 in T-cell lymphomas. For RCC and solid tumors, we're going to switch over to CTX 131. And primarily because what we saw is, while we saw a complete response in RCC with CTX 130 and we saw anti-tumor activity across a number of patients, what we did observe is as we took the cells out, that these cells were exhausted in the tumor microenvironment. And that's because solid tumors create, through TGF-beta and other signaling mechanisms, create sort of this mechanism where the tumors protect themselves and are not attacked by the immune cells. And so CTX 131 overcomes some of those signaling factors in the microenvironment that exhaust the T cells. So we're going to move to CTX 131 forward in solid tumors. And that would be pretty exciting if we have a product in solid tumors, because so far all the data for CAR Ts have been in heme malignancies.
Okay. I want to take a moment and maybe hear your view on certain developments in the CAR T space. Obviously, competition is real. We have autologous CAR T players like Novartis. They're looking for ways to decrease their vein-to-vein time. They have this T-charge process that they're looking to advance. I guess how much of a competitive threat do you think these innovations are? Innovation in the auto CAR T space to players like you or Allogene or any other allogeneic cell therapy player?
I think I'm glad that there's all these efforts ongoing. But I think with auto CAR T, if manufactured centrally, there's just a limit on how much you can improve. The vein-to-vein time is never going to compress below 10 days in my mind, in spite of all these improvements, because you can shorten the manufacturing time quite a bit by changing the activation time of the T cells and all that. But you still have to release the product. You still have to ship them, the sample cells back and forth, and you can't have them off the shelf. So there's a limit. I think, of course, if bedside CAR T becomes a reality in hospital, then that changes the equation a little bit. But we think allo CAR T is the way to go to enable access. And we're talking about thousands and thousands of patients. I just don't think the autologous therapies can scale to that level. like you could do with a Rituxan, and treat 20,000 patients a year.
Okay. And oftentimes I get, inbound from investors, who are looking at the space and they ask me, do you think allogeneic cell therapies will be reserved for those fraction of patients that cannot get an auto CAR T? I guess, what does your internal research suggest, just maybe talking to KOLs or market research efforts?
Yeah, I don't think there's going to be a one winner takes all. I think there are going to be, in the academic medical centers that have good access to auto therapies, if, there are going to be patients that are going to get auto. I mean, if this tumor is not fast growing. I think even in those settings, I think what we're hearing from physicians is if patients have fast growing tumors, or if they believe the patient's fitness level is not great, or the T cells from the patient may not be, great substrate for the auto CAR Ts, they will go to allo and then take a shot with allo versus going to bispecifics. In the community settings and in larger cancer care networks, you're going to see a lot more allo. I think outside the U.S., in the European settings, you're going to see a lot more allo because they just don't have access to auto. And so overall, my belief is you're going to see more, 4 years from now, or 4 to 5 years from now, you'll see more patients treated with allo over auto. That can come in various forms. It's not in any way niche, but you're going to see many reasons why a physician may choose to do allo versus an auto therapy.
Okay. And I guess, how are you thinking about moving your programs into earlier lines of therapy, whether it's for, large B-cell lymphoma or any other indication, the solid tumor pipeline that you have?
Yeah, I think in lymphoma, that's been one of the biggest topics coming up after our ASH presentation in December, is investigators reaching out with various ideas how you can move earlier. Ideas like frontline, high-risk patients that could benefit from allogeneic therapies. Obviously, there's a second-line treatment. And interesting combinations. What if you debulk the tumor up front and then had maintenance with Rituxan or even some other agents like Monjuvi, for instance? Would that be better than exposing these patients to a continuous prolonged period of dosing with bispecifics where there's tox risk and infection risk and everything else? I think those ideas are coming up. I think what we want to do is, as we get our registrational trial going, have further discussions with regulators to understand the bar in each and how they view it in terms of trial design, and then provide guidance around that. In solid tumors, again, in RCC, our investigators always remind us, even for axi-cell approval in frontline, the CR rate was was like 10%. You don't really see CRs, even in frontline settings in tumor, in malignant RCC. So the bar is not that high there. And if you can show CRs and a set of PRs in these indications in patients with 131, I think it's going to be a major product.
Okay. I think we might have to cut it off here, but any last minute thoughts on what investors should expect from CRISPR? I know you have obviously a lot of interesting developments on the non-IO programs, but anything on the IO front that investors should keep in mind for the next 6 to 12 months?
I think, look, I think what I've realized is, investors, especially the specialist investors, will go in waves. There's a lot of excitement around something, all of a sudden the new flavor of a different cell type, and people then will wax and wane. And for us as drug developers, we have to have a fundamental thesis and persist. That's how you get drugs approved and on the market. There were times in the last 20 years people were very negative on daratumumab and saying there's no place for this in this treatment landscape, and now it's a multi-billion-dollar drug. There were other places where people underestimated the safety risks versus efficacy and that balance. And what you see is people in the community settings take a very balanced view of that ratio of benefit to risk for patients. And the other thing is, it's not static. It's a dynamic environment. Allo CAR Ts are going to get better and better and better, whereas there's a limit to the improvement of auto and bispecifics. And as a modality, I think allo CAR T will be a major segment of the cancer market in the next 10 years. And we hope to be a leader in that space. So we're quite bullish. We're going to continue investing. And I think 23 is an important year for us as we move forward, hopefully not 1, but 2 registrational trials forward, and have data for our next gen CAR Ts, which would, kind of show us the way forward in terms of where we want to engineer the cells and where we want to go. So we're quite excited, and our entire company and everybody working on these programs are quite excited, and so are the investigators.
Okay, fantastic. All right, well, that was a good fireside chat discussion. It was great to have you here, Sam, and thanks everyone to the audience for tuning in.